We are constantly told that aging well means maintaining physical strength, and maintaining that strength requires one primary fuel source: protein. The official recommended dietary allowance sits at 0.8 grams per kilogram of body weight. But for individuals over 65, clinical guidelines push that number higher, advising 1.2 to 1.6 grams to combat sarcopenia and physical frailty. Biochemical data suggests a conflicting reality. Large-scale human studies find high-protein diets correlate with an increased risk of diabetes, cancer, and cardiovascular mortality. In controlled laboratory settings, a low-protein diet, independent of calorie count, results in a 34.9% increase in Maureen lifespan. This creates a fundamental tension in geriatric medicine. The protein levels required to maximize muscle mass are the same levels that correlate with accelerated cellular aging. Protein restriction is a distinct biochemical signaling state. It is not a synonym for malnutrition. In fact, laboratory subjects on these diets often increase their total food intake while still seeing improvements in metabolic health. Protein-restricted organisms exhibit 6 hallmarks: improved metabolic health, altered nutrient sensing, reduced senescence, preserved mitochondrial function, epigenetic modifications, and delayed frailty. This entire web is activated by an internal cellular surveillance system. Specialized sensors detect amino acid drops and initiate a defensive response. Understanding longevity requires us to look past total protein counts and map the individual amino acid sensors that dictate these systemic changes. To see how a cell detects this scarcity, we have to drop down to the level of individual kinase cascades. When dietary amino acids are in short supply, the transfer RNA molecules responsible for building new proteins remain empty or uncharged. These uncharged tRNAs bind to the stress-responsive kinase GCN2, halting protein synthesis. Simultaneously, this scarcity inhibits the nutrient sensor mTORC1, removing the brakes on autophagy. The cell begins intense maintenance, clearing out misfolded proteins and dysfunctional organelles. By shifting the ratio of these two sensors, GCN2 up and mTORC1 down, the cell prioritizes long-term repair over immediate expansion. This amino acid sensing happens primarily in the liver. But how does a localized cellular reaction coordinate an anti-aging response across an entire organism? The liver broadcasts this signal using an endocrine hormone called FGF21. As these particles reach white adipose tissue, they trigger thermogenic beiging. Adipocytes shift their function, increasing energy expenditure and burning stored lipids to generate heat. FGF21 levels correlate with significant geroprotective effects, including the reduction of circulating triglycerides and the active suppression of cellular senescence in the liver and kidneys. The necessity of this hormone is absolute. Genetically modified mice lacking the FGF21 pathway see no increase in lifespan when placed on a protein-restricted diet. Dietary longevity is not the result of simple cellular starvation. It is an active systemic rewiring driven by this hormonal broadcast. This raises a critical question. Must we restrict the entire protein molecule to trigger this broadcast, or can we isolate the effect to specific components? On this grid, we trace the restriction phenotype to the essential amino acids. Methionine serves as the universal methyl donor here.
Restricting it forces the body to adapt its epigenome, upregulating specific histone modifications to preserve a youthful genetic landscape. Just like total protein restriction, cutting methionine alone is sufficient to induce FGF21 and trigger a broad metabolic improvement. By targeting specific essential amino acids, it is possible to biochemically simulate a highly protective state without reducing the total volume of food consumed. Next are the branched-chain amino acids, a group traditionally marketed for their ability to stimulate muscle protein synthesis. Leucine activates mTORC1, but restriction data is inconsistent, sometimes causing fat accretion. We see clearer results with its counterpart, isoleucine. Restricting this single molecule increases lifespan by 33% and lowers frailty in aged mice. Isoleucine acts as a specific regulator of aging pathways, whereas general BCAA modulation remains a much less precise intervention. We must also account for the biological dark matter of longevity nutrition, the non-essential amino acids. Because the body synthesizes these on demand, dietary levels were traditionally considered irrelevant to aging. But look at arginine. High dietary doses of arginine stimulate the mTORC1 and S6K1 pathways, which in human endothelial cells correlates with accelerated markers of cellular aging. Even when a compound is non-essential, providing it in massive dietary excess can actively interfere with a cell's maintenance and repair protocols. The nutritional paradigm shifts again when looking at another group of non-essential amino acids. For these compounds, the evidence points toward targeted supplementation. Increasing dietary glycine, serine, and proline clearly extends health span across species. Glycine serves as a critical substrate separatus ion, the body's primary antioxidant. Increasing its availability helps scavenge reactive oxygen species and preserves cellular integrity. This logic is supported by studies on dietary collagen. Being exceptionally rich in glycine and proline, collagen supplementation mirrors these exact anti-aging and health span extending effects. The data suggests a bipartite approach, the removal of harmful signaling triggers, alongside the fortification of building blocks used for cellular defense. Based on all of this complex biochemical reality, what does a scientifically optimized human diet actually look like? We find a historical precedent in Okinawa. Their traditional diet was low in overall protein, averaging only 9%, and derived 80% of its calories from plant sources, which are naturally lower in methionine and isoleucine. As this graphic illustrates, their consumption patterns closely align with the molecular targets identified in modern precision nutrition. This theoretical ideal features a low overall protein intake, with restricted levels of methionine and isoleucine, balanced by fortified levels of glycine and serine. This highly specific profile stands in contrast to the generalized recommendation of 1.2 grams of total protein per kilogram currently pushed on older adults. Translating these models to humans requires careful clinical management. The immediate risk of physical frailty must be balanced against the long-term data supporting systemic health span extension. Dietary protein is no longer understood as a monolithic macronutrient. Our ability to influence health span now depends on the precise modulation of individual amino acid molecules.